Thanks for being here. So I want to start out by crediting my co-author, Lena Arvalo, who's an amazing postdoc who did all the work I'm going to tell you about today. So the genetic basis of postzygotic reproductive isolation is characterized by two features that are prevalent enough to be referred to as rules. And the first one is the observation that was made by Haldane that when one sex of hybrids is inviable or sterile, it's the heterogametic sex. And the second feature that really follows from Haldane's rule is that there's often a disproportionately large contribution of the X chromosome to these hybrid phenotypes. So there are a number of explanations for this large X effect, but the one that's most broadly applicable is that in the heterogametic sex, alleles on the X chromosome that act as recessive incompatibilities are always going to be exposed, whereas they're masked in the homogametic sex. So today I'm going to tell you about a cross between two species of mice, the house mouse, Mus musculus domesticus, and the Algerian mouse, Mus spreadus. And I'm just going to call them domesticus and spreadus for simplicity. So there is hybrid male sterility in this cross, but I'm going to talk today about an inviability phenotype that I think is equally interesting. So in this cross, as in a number of other crosses in males, there's this really unique phenotype where there's asymmetry in abnormal growth patterns across reciprocal directions of the cross, so that in one direction of the cross, the offspring are oversized, sometimes to the extent of being disturbingly so, and in the other direction, they're undersized. And the same is true in the cross that we work on. So in the cross between domesticus and spreadus, when the paternal species is the house mouse, domesticus, placenta and embryos are oversized, and when the father is spreadus, placentas and embryos are undersized. And I should say that in all of these crosses, these fundamental differences in size start with differences in size in the placenta. And so this is generally considered to be a signature of disruption of genomic imprinting. So imprinted genes are really interesting for a number of reasons. They're autosomal genes, but they have this unique monoallelic pattern of expression where one allele is expressed and the other is suppressed or largely suppressed by some sort of epigenetic mark, often, as I've shown here, DNA methylation. But the other reason that imprinted genes are really cool is that about half of them are expressed from the paternally inherited allele and the other half from the maternally inherited allele. And this is, in fact, what makes them strong candidates for these reciprocal opposite patterns in hybrid crosses. But the other really unique thing about imprinted genes is that they're highly, highly expressed in the placenta, and where several of them are critical to development and growth. And so this makes them altogether really strong candidates for these phenotypes and hybrids. So we're interested in the genetic basis of these growth abnormalities in hybrids, and prior work on this cross suggested that imprinted genes were indeed involved from some candidate gene studies. And interestingly, there's also evidence that the two rules of speciation are followed in these placental phenotypes, so that they're more severe in hybrid males relative to their sisters, and there's also evidence for a large effect of the X chromosome on these placental phenotypes that are more severe in males. And the reason that this is surprising is that the placenta is unique. So in female rodents, they selectively silence the paternally inherited X chromosome. And so the effect of this is that both sexes are effectively hemizygous for the maternally inherited X. And so therefore you would not expect in this tissue to see Haldane's rule of the large X effect. Okay, so the first thing that we did was to make sure that we could recapitulate the results that others had already shown. And so we measured placental efficiency, and this is just placenta weight over embryonic weight. And we found that indeed we could find the same patterns. And I should say that all the work I'm going to talk about today is just in the direction of the cross where the placentas are undersized relative to parental species. And that's indeed what we find. And we also find that the hybrid male placentas are significantly reduced reproduced relative to their sisters. And we've also found that resorbed embryos are male biased, directly linking this phenotype to inviability. Okay, so the questions that we're interested in is what's the genetic basis of these hybrid growth abnormalities, and like I already told you, imprinted genes are kind of strong a priori candidate. So most of what I'm going to talk about today is expression. So the first thing to do is to see if the imprinted genes are misexpressed in these hybrids, and if so, is there more misexpression in males than females? But we're also interested in why it is that in this tissue where you wouldn't expect it, the phenotype is more severe in male hybrids. And it makes sense to at least first look at the X chromosome and simply ask if there's a lot of misexpression on the X and whether there's more of it in males than in females. So there's a lot of stuff on this slide. I'm not going to say it all. What's most important is that we did 3 types of crosses: the interspecific cross with a domesticus mother, a spreadus father, and then 2 intraspecific crosses. And we collected placenta in late gestation, and then we used that to sequence transcriptomes to look at differential expression, and also we used reduced representation bisulfite sequencing to look at genome-wide patterns of DNA methylation. Okay, so the first question is, are imprinted genes misexpressed in hybrid placenta? And so in the placenta, we used, there's a lot of debate over how many imprinted genes there actually are, but so we used a set of 135 genes that are validated as imprinted in the placenta. And so this is obviously a small subset of all the genes that are expressed in the genome. So I thought it'd be disingenuous to show you just the genes we're interested in. So first, what I'm just going to show you is differential expression across the whole transcriptome. And these are volcano plots. On the X is just a measure of how different expression is for each of these points as a gene in these pairwise comparisons between hybrid placenta and domesticus placenta, hybrid and spreadus, and then y-axis is just how significant these differences are. So there's lots of differential expression. All the red is significant. But among this sea of red, there's some blue dots that maybe you can see, and these are the imprinted genes that are differentially expressed. And so we find that, in fact, just in differential expression, that imprinted genes are overrepresented. But what we're really interested in is misexpression. And so for genes to be considered misexpressed, we require that they be transgressively expressed outside of the range of both parental species. And so there are about 450 of these genes that meet this criteria, of which 7 are imprinted genes. So right away, this tells us that not all imprinted genes are misexpressed in these hybrid placentas. So now I'm going to focus in on the imprinted genes that are misexpressed at the level of the whole transcriptome. Okay, here they are. So this is the number that are, now I'm splitting by sex. So this is in males and this is in females. And I'm not going to read all the stuff in these tables. I just want to make 3 points. First is that there's clearly more misexpression in males than females. Second, imprinted genes tend to be found in clusters in the genome, and we have one cluster in particular that's on chromosome 7 that's really overrepresented in this group, that there's a lot of misexpressed genes in this cluster. Third thing is that we find differential methylation in association with this. So imprint control elements are regions that control the expression of the genes in the cluster, and this is differentially methylated. And we find differential methylation in the promoters in males. So I'm just going to focus on 2 genes that we think are particularly good candidates for a contribution to these placental phenotypes in hybrids. So here they are, ASCL2 and FILDA2. And so the hybrid phenotype in terms of expression is for ASCL2, there's transgressive underexpression in males only. And then consistent with the direction of expression in males, we find hypermethylation in the promoter relative to female hybrids. And then the imprint control element is hypermethylated in this whole cluster of imprinted genes. FILDA2, the same thing, but in the opposite direction. So there's transgressive overexpression in both sexes, but this, Again, more so in males than females, and again, consistent with expression hypomethylation in the placenta in the promoter region in males. So there's actually been a lot of work done on these genes in lab mice, and they play a really critical role in placental development. I'm just going to give you the short version of all this. So if you knock out ASCL2, it's embryonic lethal. If you reduce the expression of ASCL2 twofold, you get basically the same general phenotype that we see in our cross. And in our cross, it is the expression is reduced. Same is true for FILDA2. So if you overexpress this gene, which is what we see in the hybrids, you get placenta and embryonic growth restriction, the phenotype that we see. So this is cool. This is obviously just associations at this point, but we think that these are exciting candidates to go forward with. However, this in no way explains why males are worse off than females as hybrids. So now we've seen that the placental phenotype is more severe in males, and also the gene expression phenotype is more severe. So to start to deal with this, we maybe kind of naively asked if there was generally overexpression or underexpression on the X in males. And again, just to sort of keep things honest, I'm just showing you first the differential expression. So this isn't necessarily misexpression in the pairwise comparisons, hybrid versus domesticus, hybrid versus spreadus, now split by males and females. And so the only point I want to make here is that there's no obvious pattern of a big difference between males and females, or up or down regulation on the X. And so, in fact, if we ask this question, whether X chromosome genes that are misexpressed are overrepresented on the male X relative to the autosomes, the answer to this question is no. So at this point, we thought, okay, well, just because in regular female rodent placenta, the paternally inherited X chromosome is silenced, maybe this isn't entirely the case in these hybrid females. Maybe there's some sort of rescue effect from leaky placental inactivation of the paternal X. And so what we did was we looked at allele-specific expression on the X chromosome. This is just in hybrid females. And what we find is that in general, there's a strong bias towards the maternal allele on the X, the domesticus allele, which is what we expect. However, there's a pretty large tail out here where we get both biallelic expression and, even more interestingly, expression that's biased towards the paternal allele. So there's a lot of interesting stuff here, but just as kind of a first pass, what we did was we looked for the overlap between genes that are expressed from the paternal allele in females and genes that are transgressively misexpressed in males. And there's actually just one gene in there, but it's a potentially interesting candidate. It's a chromatin modifying protein. So at warp speed, what have I told you? We found that a subset of imprinted genes are misexpressed, and we find two that are really strong candidates for the phenotype. We don't know yet why it is that hybrid male are worse off than hybrid females for this phenotype, but we speculate that there's a possible rescue effect of leaky paternal X inactivation in hybrid females that makes their phenotype less severe than that of hybrid males. And in general, we think this is cool data because it shows that dysregulation of epigenetic processes, both in regulation of imprinted genes and X inactivation, can contribute to postzygotic reproductive isolation in mammals. Thank you.